Hi there, everyone. My name is Prerak Juthani. I'm an MD MBA student here at Yale, and this is part of our daily journal club where I'm going to talk about the most interesting case reports, reviews in medicine. Obviously, I'm not an expert, but I love that I can use this channel as a way to share my enthusiasm for medicine with others, and in the process, hopefully help you learn something and also help myself because I'm able to teach something about it. Today is a very interesting topic. It's about sesquizygotic twinning. Just by the name, you can tell that I clearly haven't heard of this before. Not many people have. It's a very unique phenomenon, but I'm excited to talk about it with all of you because it is a very educational case. New England Journal of Medicine, feel free to look that title up. It's going to be linked in the description below. Let's just start with the basics. Monozygotic twinning, and there's monozygotic twinning, and then there's dizygotic twinning. What does this mean? Well, when you have twins, they can come from one egg that splits into two, and that usually is called a monozygotic twin because that's one zygote, one fertilized egg with a sperm, that is then leading to two different individuals. These individuals share 100% of their DNA because, again, they came from the same egg and sperm. They just happen to split apart and form two different entities very early on. Dizygotic twins are exactly what you would think. There's two zygotes, which means the mom happened to release two eggs that got fertilized by one sperm each that led to two zygotes. These two zygotes are about as similar of siblings as you may be with your brother or sister. They're 50% similar on average, and these are known as fraternal twins. You may have heard that. Sesquizygotic twins, on the other hand, are in between. They're in between totally identical twins and totally dizygotic twins. So they share between 50 to 100% of their DNA, and these are twins that you might be interested in how they form, and we're going to go over that. Before we go further, it's important to understand the basics, right? So as I said, this is monozygotic twins. One egg gets fertilized by one sperm. Depending on where they separate, you're still going to get identical twins, but sometimes they share a similar chorion, which is the outer layer, but they have two amnions. Sometimes they have the same chorion and the same amnion, as you see here. Sometimes they have two chorions and two amnions, as you see here. Also notice how this can be very different. On the other hand, if you have dizygotic twinning, which is two different eggs fertilized by one sperm each that leads to two different embryos, this is when you can actually have entirely two different babies. Notice that in the identical twin case, even when you have different levels of amnion or chorion, they're always usually going to be the same gender, right? If something shares the same chorion, usually you assume it's an identical twin, and if it's an identical twin, you will also notice that they have to be the same gender, right? You can't have identical twins without the same gender. So now let's jump into this case. In this case, a woman was pregnant, and first trimester ultrasound showed a 28-year-old female who had a monochorionic diamniotic placenta at 6 weeks of gestation. So that kind of makes you think monochorionic diamniotic, kind of here. So that's what made them think that this was a monozygotic twin, because there was one chorion. And so now, when they did that, they then said from 14 weeks, when they then did the ultrasound, they actually found that one fetus was male and another fetus was female. This is something that you don't expect, because as I said, when you have a monochorionic diamniotic twin, you usually assume it's a monozygotic twin, and monozygotic twins have the same 100% similar DNA and therefore cannot be different sexes. So this is what blew their mind. They said, how can these two entities have the same chorion and still be different genders? And that's ultimately what led them to investigate what was going on, and they did a genomic analysis of each of the twins. When they looked through everything, they actually found that the DNA from each of these people, these little babies that came out, they shared, this boy and girl, shared 100% of their maternal DNA. So this boy and girl twin had the same maternal DNA, but the paternal DNA was different, and they shared about 77% of it. So the real question is, how did all of this happen? And this is the part about this whole case study that's really interesting, because you have to think about hypothetically how this could happen and rule out other ways that this can happen. Obviously, I'm not an expert in this. I'm just trying to show you why I found this to be so fascinating. But what they ultimately found was that there likely was a dispermic fertilization of one maternal egg. And that ultimately led to a zygote that had multiple paternal genomes that segregated independently This is called heterogenetic cell division, and I'm going to show you a picture of this later. And they also found that dispermic fertilization can lead to different embryos that share the maternal DNA, but the paternal DNA is going to be different in each of them. So now we're going to talk a bit about how this happens, and I think this is really, really fascinating stuff. So you may know sperm are formed by meiosis, meiosis 1 and meiosis 2. Meiosis 2 leads to 4 haploid sets of sperm that come from one 2N2C progenitor sperm. Similarly, females also have meiosis happening with their eggs. The meiosis egg is usually arrested in meiosis 2 until it gets fertilized and then undergoes the second round of division. Notice how this MCAT knowledge is still around. So what they think happened here is that maternal egg was sticking around. 2 sperm fertilized it at the exact same time. So notice that this is the paternal genotype from one sperm. This is the paternal genotype from sperm 2. This is the maternal one. And then what they found is that they think there was a trispindle apparatus, right? And this led to 3 different entities. One that had the maternal DNA and the sperm from and the DNA from sperm 1. Another that had the maternal DNA and the paternal DNA from sperm 2. And another one that had the paternal DNA from sperm 1 and the paternal DNA from sperm 2. They have found by studies done in the lab that when you have 2 sets of paternal DNA come together, it's actually not viable. And so this one right here actually didn't proceed further past the original stage. Only this one and this one proceeded. And that's ultimately what led to this monochorionic diamniotic twinning. But notice that they both share the same maternal DNA, but sperm 1 and sperm 2 are 2 different sets of sperm, which is why you can get 2 different genotypes, which is absolutely fascinating. So now all this to say, these 2 offspring have the same maternal set of DNA, and their paternal sets are 77% identical, which makes them 80% identical in their genotype, which is why they're known as Sesquizygotic twins, because they don't share 50% of their DNA, which would make them Dizygotic twins. They don't share 100% of their DNA, which would make them Monozygotic twins. They share something in the middle, which is fascinating, interesting stuff. And this is the theory of how they think it happened. If you want to read more, I strongly encourage you to do so. I'm going to link this whole article in the description below. If you enjoyed this, please drop a like, comment, share, subscribe, and definitely going to have more journal clubs in the future, especially as I start residency and continuing wanting to share my insights with all of you. So thank you so much for watching, and I hope you'll join me very soon in the next video. Peace.